Well, thank you everyone for attending my talk today. I'd like to thank Marcy for inviting me, and then also Aaron for helping me set up today. So I'm going to be presenting on some work that my lab did a few years ago, as well as just some general research that we do. So my presentation is called From Proteins to Nanoparticles, Domain-Agnostic Machine Learning for the Nanoscale. And I'd like to start with some basic motivation. So there are two main problems that our research is sort of motivated by. One is the main sort of area is this issue of antibiotic resistant bacteria, which I'm sure everyone is aware of. Basically, as we develop new antibiotics and use these antibiotics incorrectly, bacteria become more immune to these antibiotics that we develop. And so the number of antibiotic resistant infections and deaths has been increasing significantly over time. And also, we're sort of motivated by this issue of pandemics, which we've all had to deal with recently. And there's sort of a prescient quote from in 2019 from the Worldwide Threat Assessment community that they said that we anticipate there will be more frequent outbreaks of infectious diseases because rapid unplanned urbanization, prolonged humanitarian crises, human incursion into previously unsettled land, expansion of international travel and trade, and regional climate change. So these are all things that are not going away and are only going to become more common. And so we sort of want to come up with some way to help address this issue. So what is our solution? So the broad overarching idea is that these are both problems that can be addressed using nanochemistry or various kinds of nanoparticles. And so these are just some examples of nanoparticles that you could use. And basically the definition of a nanoparticle is anything that's on the order of 1 to 100 nanometers. And so as you can see, they can have varying structures, but their definition is basically just based on their scale. And so you may ask, why do we think that nanoparticles are interesting? Why do we think that they're useful for these sorts of applications? And the main reason that we think that they're useful for dealing with antibiotic resistance is that there are many different ways that nanoparticles can attack bacteria. And because there are many different ways, these bacteria are less likely to be able to develop resistance to any individual nanoparticle because there are just too many pathways being attacked at the same time. So in this graphic, we can see that there are multiple ways that this nanoparticle can enter the cell. It can enter through the membrane or it can enter through pores. And then when it's in there, it can attack, this is respiration. It can have oxidative, reactive oxygen species that attack various structures within the bacteria. And then it can also mess with the signaling pathways. So this is just an example of one kind of nanoparticle and the sort of multi-faceted way that it can attack bacteria. And then, so more broadly though, the main sort of things that we're interested in with nanoparticles is their ability to interact with proteins. So here we see a type of nanoparticle called a molecular tweezer that's binding to a very specific site on a protein. And then we're also interested in the ways that nanoparticles can be used for delivering, I guess you could call them warheads or payloads to cells or bacteria. This is an example of an mRNA vaccine, which takes the mRNA and it places it inside of a lipid nanoparticle, so that way it can survive through the bloodstream, survive in the host and then be taken up by the cells to actually produce the antibodies. And so, yeah, in our lab, we mainly focus on nanoscale chemistry as sort of a general concept. This is an illustration of some nanoparticles in like a plasma. These are specifically silicon and hydrogen, I believe. And so we look at like the growth of these nanoparticles in plasmas, and we also look at like the nanoscale interactions that happen between like proteins, for example. So these are two extremely different systems that both can be sort of classified as nanoscale interactions just because of the length scale that they operate on. But then some other work that we did that's maybe more directly related to the motivations we discussed before is this idea of protein and nanoparticle interactions. So on the top row, you can see this is so in a bacterial or in a when you have many bacteria that live in an environment together, they'll cooperate to develop something called a biofilm, which is basically like a protective barrier that they make that protects them from antibiotics or like drying out or things like that. And the way this biofilm is formed is by excretion of proteins that then aggregate to form these long sheets that then create sort of like a like water retaining polymer. So we have a paper that basically shows that if you take this biofilm and you add a bunch of graphene quantum dot nanoparticles, then these nanoparticles will end up binding to the ends of these, I believe they're PSM alpha proteins, which prevents them from aggregating. And I probably should have included an image here, but it basically makes the biofilm break down. So that way you can actually more directly attack the bacteria, which would have previously been protected. Let's see, nanoscale chemistry that we cover also includes like the interactions between proteins and like lipid membranes. So like here is the COVID spike protein, and we had a paper on how this interacts with the cell membrane. And then also we're interested in the effects of combustion and the nanoparticles that are produced by combustion and how these can enter or affect cells. So here we see an example of a carbonaceous nanoparticle that is outside of the cell, and then over time how it moves through the lipid bilayer and sort of how it enters the cell and what its effect is. So there are a lot of works that have sort of focused. Yes.
Just a quick question.
Yeah.
When you talked about biofilms.
Yeah.
Is the nanoparticles that you're talking about able to disrupt a biofilm as it is forming, or can it go after a biofilm that has already been formed?
I believe the paper is on biofilm formation. I wasn't on this paper. This was before I joined the lab, but my understanding is that it's on biofilm formation.
Okay.
And I can, if you'd like, I can look into it and see, get back to you.
All right.
Yeah, so there are a couple existing works that focus on things that could be considered nanoscale interactions.
There is actually a raised hand online.
Yep.
From Angela, so if you give me a moment, I can let her talk to you.
Okay, yeah.
Angela, I think you should be able to talk.
Yes, can you hear me?
Yeah.
Yeah, I just want to go back to the last question I received. So what we did in the paper was basically show that nanoparticles interfere with the formation of those bacterial amyloid fibers that you find in biofilms, but we're also able to disrupt the fibers once they are formed. And so what you see here, I think in the slide, is basically the fact that the presence of nanoparticles does not allow those peptide to keep growing and form fibers. But we also did some analysis in which once you form the fibers, the nanoparticle actually disrupt the structures. And so basically breaking down into small peptides. Thank you. Sorry.
Thank you.
Yeah, so there are a few works that already try to address issues that could be considered nanoscale interactions. Maybe one of the most well-known ones is this idea of protein folding or protein complexing. And there was a very, very famous one that came out a few years ago, and then they had a new, maybe in like 2016, then they had a new version that came out a few years later, and they've sort of been updating it since, but that's AlphaFold, which is achieves very, very good predictive accuracy on taking some protein sequence and actually determining what structure is going to be formed using that sequence. And then this approach has also been used to predict protein complexes. So here on the right is an example of, I believe, 4 proteins that, well, 4 or more proteins that have been, I believe they took the linearized sequence, they folded them, and then they predicted how those proteins ended up binding to each other in a complex. And this is also using AlphaFold. And then there are some methods that are focused specifically on protein complexing, where they basically they assume that you have the unbound structure, and then they're sort of operating based on that. Yeah, there's also something called ligand docking, which is basically where you have some protein and you want to figure out where on it like a small molecule will stick to. This is, I believe this is typically how like drugs are developed, is you find some site on a protein that you want to modulate using some drug, and then you try to identify what kind of drug will bind there, or screen through a series of drugs to see if they will bind to that site. And this is also using AlphaFold. And then there are various methods that are developed specifically for this. I believe this one is called Coach, which is basically like a consensus method that takes many different types of models and sort of combines them, so it can make predictions for like binding of DNA to proteins, or peptides or ligands to proteins. And then sort of related to this is this idea of pocket detection, which is similar to ligand binding, but you don't know what drug you actually want to bind. You just want to find like all the possible binding sites on a protein or some small molecule. So you basically take your protein and you try to identify some region where some hypothetical ligand could stick. And there are a couple different ways of doing this. The first two are using sort of analytical methods based on local neighborhoods. Or second, I believe, is based on just trying to find like relatively concave regions of the protein. And then the last method is sort of like a neural network based approach, which tries to look at some local neighborhood of the protein and use like a machine learning model to make this prediction. And then sort of the final approach that people will take is something called protein interface prediction or recognition. And basically the idea is like you try to predict, it's more or less like pocket detection, but you're trying to find all the places where some hypothetical protein could bind. And then you try to sort of find the union over all of those possible binding locations. And you get some prediction kind of like this, where white is like the not binding region. I believe red is the correctly identified binding region. So there are a lot of methods that have been developed to do this sort of thing, but they all have some they all have some shortcoming in one way or another. One issue is that ligand pockets are too small for nanoparticles. So if we look at like the ligands, in some settings, they can find like DNA binding locations, but generally they're meant for small molecules that are maybe like tens of or dozens of angstroms across. So but what we're interested in are nanoparticles that are in some cases basically the size of the protein itself. Then also there's sort of an issue that short range nanoparticle and protein chemistry differs. So we may be interested in a nanoparticle like this Buckminster fullerene. I think this is actually one that we tested on in our paper. But when you're dealing with proteins, you have very different sort of local chemistry. So these deep learning methods that are based on very local neighborhoods may not work particularly well. And then a lot of methods for making these interface predictions are not partner specific. Like I said, with the pocket recognition and the protein interface recognition, basically you're given a protein and you're trying to determine where on that protein a nanoparticle will bind, as opposed to something more specific like protein interaction prediction, where you're given both the you're basically given the two proteins and you're trying to determine where they'll bind and or if they'll bind and where specifically that binding will happen. So the questions that we wanted to address with this work is, can machine learning predict protein nanoparticle complexes in general, not for a very specific class of nanoscale interactions? And then sort of a precursor to this is, why is there so little work on protein nanoparticle interactions? Because all these methods were kind of focused on either protein-protein or protein ligand, with maybe like protein DNA or something like that added in. And so there are a couple of main reasons. One is that the nanoparticles sort of by definition have very diverse chemistry, because it's sort of defined on the length scale and not so much the composition. There are many different types of nanoparticles that you could have. Like you could have like lipid nanoparticles, you could have like metal nanoparticles with shells. You could have, as I mentioned before, you can have like these graphene quantum dots, which are completely made of carbon. You can have polymers. So it's very difficult to develop some general method. Also, nanoparticles have like a very large structure. So here is some, just for illustration, like a caffeine molecule next to some lipid nanoparticle for mRNA delivery. You can see that like the lipid nanoparticle is very large compared to the caffeine, meaning that if you have some algorithm that's designed, like some machine learning algorithm or something, you have a lot of data that you need to process, but you have not a very large number of samples, which is the issue with the small data sets. Like if you look at the protein data bank, which is sort of a set of all the known protein structures, then you have hundreds of thousands of protein structures. But if you look at, for comparison, if you look at the largest like structural nanoparticle data set that's sort of been collected, I believe this is Venus, then there are only hundreds of nanoparticles for comparison, and most of these are specifically gold nanoparticles. In the other situations, you have like tens of examples. So if you're trying to like train some model, you don't have a lot of data to go on. And so the sort of our answer to number one is, why is there so little work on protein nanoparticle predictions? Because there's much less data for nanoparticles than proteins. So so proteins are basically just an easier thing to approach from the beginning, and they're also much more structured. So it's reasonable to start there. But then that brings up maybe a second question, which is, can protein data enable protein nanoparticle predictions? Like, in other words, if we don't have a lot of nanoparticle data, is there some way that we can use data that we have a lot of to help in the situation where we don't have a lot of data? And so basically, the more abstract problem statement is like, we have some problem where we want to predict, maybe like, will these proteins interact? Will these proteins not interact? Or sorry, will a protein and nanoparticle interact? Will protein and nanoparticle not interact? And we don't have a lot of like data points to train some model on. So if we want to train a model, we can end up with all sorts of different solutions that work on our very small data set, but are not actually generalizable to the real problem that we're interested in. So there's this thing that we can do. But what we do have is, we have a large data set of protein interactions, which is not the same, it's slightly different. But the idea is that if we can sort of describe these proteins and nanoparticles in the proper way, then maybe some model that we learn for the nanoparticles will also be relatively good for the protein nanoparticle interactions. And so this is some area of machine learning called transfer learning, where you train on some large data set, try to evaluate on some related smaller data set. And yeah, so the idea in our setting is, we take the protein data and we basically train some machine learning model on pairs of proteins and predict how and where these proteins will bind. And then we are going to take some protein and nanoparticle combination, and we're going to try to predict where these proteins and nanoparticles will bind without doing like any additional training. We just test it on the protein nanoparticle interactions. And so, like I said earlier, the local protein nanoparticle chemistry can be very different. So why do we think that this should work? And the idea is basically that, at least in the setting of organic nanoparticles, if you look at sort of local regions of these nanoparticles, then this is sort of similar to the idea of taking like a coarse-grained structure of protein, which is here basically each of these beads represents the chemistry of some local region of the protein. And then, even though the chemistry is not necessarily the same, if you take this like coarse-grained representation and then do some sort of and then sort of compute like a more the more like wide range properties of the chemical structure, then you may be able to do some transfer learning between these. So basically, in our paper, what we did is we took 230 pairs of 230 sets of protein complexes, and we trained a model on this. And then we took 21 protein nanoparticle pairs, and we didn't do any training on this. We just did some evaluation, and we took both of these data sets and we did some coarse graining on them. So we basically tried to look at some, I guess, local chemistry, and then we computed some features of each of these regions instead of looking at like atom level features. And yeah, so that's the approach that we took. So for coarse graining, basically what the idea with that is that we take our atomic structures, which are maybe high dimensional, and they can be noisy, like you can have a lot of thermal noise. And the chemistry is maybe on the atomic scale very different. And then you sort of compute some local, you basically find some local regions that are less prone to noise and are possibly share are more likely to have similar chemistry than if you would just look at the individual atoms. And so this isn't an image of our approach. This is sort of an image of the algorithm that we use applied to some different setting. I just found this on the internet. But basically what we do is we run this algorithm that takes some bunch of points and then it finds some smaller number of points that more or less describe the same volume. And then we basically use this approach to identify regions of to identify like the centers of each of these spheres in our proteins and our nanoparticles, or proteins that are nanoparticle structures. And then to capture the local chemistry in a way that is able to be transferred between proteins and nanoparticles, we basically took some structure and like took some individual bead from that structure, and then we computed some list of properties that we think will probably be consistent across protein and nanoparticle structures. So the CPSA is like the charged particle surface area. WIM is some list of like features that are basically supposed to be invariant to they're so I'm not quite familiar with these because this is not my specific part of the project, but these list of features I'm told are very similar between proteins and nanoparticles. And we chose these with the knowledge that we were working on organic nanoparticles, but this feature list could be adjusted if you knew that you were going to be working on some other kind of nanoparticle, like possibly like a metallic nanoparticle or some like lipid nanoparticle. And so this is sort of to compute like the local chemistry for a very specific region. But if you just look at these local regions, then you may not actually collect the information or use the information about the sort of broader surrounding region of the nanoparticle, which will make it difficult to make accurate predictions because you won't sort of know how this region behaves in context. And so what we do is we perform some weighting of these features, basically like a weighted average, where based on some distance between two beads, we'll compute some weighted average based on this Gaussian function. And so an illustration of this is like if we have this specific bead right here, we'll take some neighbors, like for this one right here, and we'll basically compute like we'll add some features to this bead based on some weighted average of its neighbors' chemical information. And then we'll also do the same thing for some more long-range properties. In this case, obviously the closer ones will be weighted more than the ones that are farther away. And then hopefully, or at least the idea is that this sort of ameliorates the issue of focusing on extremely local chemistry, and we get more long-range interactions. And so the general approach that we do is we take these coarse-grained structures, we use this weighting approach that I discussed to compute features for each of these beads. So each of these vectors is associated with one of these beads on the structure. And then we give it to some machine learning model, which basically takes in one bead from each structure and then computes some binary prediction that basically says, like, given these two structures, will these two beads interact? Will these two beads interact? Will these two interact? And basically the idea is if we do this, then we can compute some bipartite graph, or basically some interaction prediction between all the pairs of beads between both structures. And then we can basically get some interaction potentials between them. So when we use this approach, we tried it on two things. We tried it on protein nanoparticle and protein predictions. So this is a list of previous methods that have been developed. So a lot of these are non-partner specific, meaning they're not aware of the specific nanoparticle that they're going to be binding to. And these are all developed for either protein, or I believe these are all developed for either interface detection, like for protein-protein interface detection, or for ligand pocket detection. But they all perform, relatively poorly. And then this is a method that was developed for protein nanoparticle interactions, specifically more recently. And it performs better, but it's about the same as Spider, which I believe is a protein-protein interaction prediction method. And then, so we have our method. So this is sort of an ablated version of our method with some properties, or some of its capabilities reduced, I guess. And then our approach, NeCLAS, basically outperforms all the other methods by a significant degree. And this long tail is basically due to two structures that have very different chemistry from the rest of the nanoparticles. So we think that, like, in this extreme setting, we're not training on any nanoparticles at all. But if we add just a few, like a little bit of, or a few nanoparticles to the training data set, then we may be able to sort of improve this tail performance. And then if we try the same thing on some protein data. So these are just two different ways of splitting the protein data, because when you're making your training and testing set, we were evaluating against two methods that have a different way of grading testing or training and evaluation data sets. And both these methods, PIPSP and PIPGCM, are meant specifically for protein-protein interaction prediction. And this is this other method for protein nanoparticle interaction prediction. And our method is here. So it performs, in a lot of settings, it performs almost as well as protein-protein, or methods that are specifically developed for protein-protein interaction prediction. But it can also perform very well on organic nanoparticle interaction prediction, which it was not trained to do. We just developed the features with the intention that it should be able to generalize to this setting. So one thing we did is we looked at a very specific example of where the prediction went very well. We tried to see what exactly the prediction was. So here you can see there's a this is a protein nanoparticle prediction between a molecular tweezer and some protein. And in the red is basically where it's predicted that the molecular tweezer will bind, and the yellow is where it will not bind. And basically, if we zoom into this, we can see that this is actually where the nanoparticle is supposed to bind. So our method for this specific situation is identifying correctly the binding region with significant level of precision. So then we also wanted to investigate, like, what specifically is the model learning. So we took this graphene quantum dot and we coarse grained it, and then we took this protein PSM alpha and we coarse grained it, and then we basically ran some simulation, molecular dynamic simulation, to see how the protein and the nanoparticle behaved. And what we found is that so these pink dots represent these center beads in the GQD, and the green beads, or the green dots, represent the green beads in the quantum dot. And basically what we find is that when you run this molecular dynamic simulation and you plot the contact time between the protein and these two structures in the GQD. The prediction of NeCLAS's prediction on how likely they are to any pair of like beads to interact is correlated with the actual time that they spend contacted in the molecular dynamics simulation. So basically, this means that NeCLAS is not just identifying where these the probability that these any pair of beads will interact, but it's sort of identifying the pair potentials between these coarse-grained structures, just like you would sort of use if you were developing some coarse-grained molecular dynamic simulation. So then we wanted to see if this would actually work for doing nanoparticle predictions, which was not trained on it all, it's trained just on protein data. And so we looked at two structures. These are both graphene quantum dots, and we basically used the NeCLAS predictions to develop these potentials between pairs of structures, the interior beads, interior and exterior, and then between the exterior beads. And when we've done some coarse grain simulation with these potentials, we find structures like this, which are qualitatively similar to the structures that we find if we actually run the full molecular dynamics simulation. So basically, the NeCLAS is able to identify these coarse grain potentials in interactions between two nanoparticles after only being trained on protein data. So the answer to the second scientific question, can protein data enable protein nanoparticle predictions? Well, we think that the answer is yes, if you look at the right length scale and if you choose your features carefully, so that way your predictions for protein data will extrapolate to protein nanoparticle or nanoparticle predictions. So what is the message behind this? Our approach can model many nanoscale interactions. We evaluated it specifically on organic nanoparticles and their interactions with proteins. But we've shown that the predictions that we make actually generalize beyond binding to pair potentials. And then we think that this approach will have applications in things like nanodrug design and validation, and the modeling of nanoparticle aggregation or protein aggregation and the effect of nanoparticles on those aggregates. Yeah, so future work that we can do with this is obviously we can have a larger training data set. We only had 230. We only trained on 230 protein pairs. We could add many more. We could broaden the range of nanoscale interactions to include things like metals, which we haven't done in this paper specifically. We can also look at more solvents, because everything we have is specifically soluble in water. But I guess the idea and why this is relevant to you is that if you are developing any kind of drug or anything where nanoparticle interactions are important, if you can make accurate in silico predictions efficiently using like a machine learning method instead of running expensive molecular dynamics simulations or running expensive wet lab experiments, then you have to do less synthesis and fewer computational simulations, which gives you a faster turnaround in your development process. So I guess, like I said, one important caveat is that we have not evaluated on inorganic nanoparticles to my knowledge yet, but that's sort of direction that we're planning on taking it. And then one final note that I want to make is that since this paper was published, this method AlphaFold 3 came out, which it claims is able to do protein interactions, protein ligands, and this is more general, some more general nanoscale interactions. Like here is a protein DNA interaction. However, one thing that at least I noticed when I read through the paper is that they don't anywhere, or nowhere they talk about, their ability to perform general nanoscale interactions. And they make the, caveat in their paper that they are able to, make accurate predictions for most, species in the protein data bank. They list various things that they're not able to, predict well on, but they don't make any mention of more general, nanoparticles or nanostructures. So we think this is at least one place where we still have the potential to perform better than alpha. Yeah, so I'd like to thank my collaborators, Jacob Saldinger, who's the first author on this paper, Chloe, who helped with some simulations, and Angela, who's my advisor and who's also the last author. And then I'd also like to thank Paolo Elbati, who was the third author, I believe, on this paper, and who's also very instrumental to write this. Then I also want to just acknowledge some funding that we got, and also my other co-advisor who helped a bit with the machine learning, and then the computing resources that we used. Yeah, so thank you. It's the end of that. Yes.
Yeah, I have a basically two questions.
Mhm.
So I do believe that the protein-protein interaction is mainly governed by lock and key model or induced bit model. However, regarding the nanoparticles, I think different chemistry applied like charge matter, size matter. I'm not sure, I mean, the transportability of the machine learning model is possible. This is my first question. And my second question is, how do you validate that experimentally, like cryo-EM or NMR, right through?
Right, okay, so for your first question, which was, where's my mouse going? Okay, anyway, your first question was the difference in, like,
Chemistry?
right, lock and key versus like charge. So this method, sorry, going way back, this method right here, Unified, I believe, this is a protein nanoparticle interaction prediction method. And my understanding is that in their method, they make the assumption about the lock and key. They assume that applies to nanoparticles as well. Because they use something they call geometric features, which are basically like properties of like concavity or like concave regions where a nanoparticle could bind. They don't perform as well as we do, and I believe they make that assumption that the lock and key mechanism occurs. I don't think that we do that. We just look at, I don't think we make that assumption. We look at more like physical chemical features, like we include charge as one of our features that the model takes into consideration. And I don't know specifically what the model uses, but I think it's possible that because we're not explicitly including geometric information in this, we're mainly including, like you said, like charge, that the model is learning to use this for these nanoparticle interactions. Then your other question was about
Experimental validation?
yeah, so we didn't run experimental validation, but these data sets that we used, these data sets are experimental data sets. They come from the protein data bank, I believe, which is cryo-EM generally. So like these specific structures that we tested are known. The ground truth is known. So we didn't test it on an unknown structure and then validate ourselves. We looked at things that were already known. Does that answer your
Yeah,
question?
thank you.
Okay, yeah. Yes.
Again, a couple of questions,
Yeah.
sort of speculative ones.
Mhm.
If you have a number of these nanoparticles that bind specifically to different proteins,
Mhm.
is it possible by looking at that interaction to identify the protein, an unknown protein that you have in front of you? Or is that not quite there yet?
So your question is, given multiple nanoparticles that bind to something, but you don't know what it is, could you determine what that thing is?
Yes.
So this method, I don't think so with this method, because our approach is partner specific, meaning that let me get to the region. Sorry, we assume that you know both of the structures, and you look at both, like you look at the regions of both the protein and the nanoparticle. So I think our approach would apply to the situation where you know what the nanoparticle or what the protein is already. I think you could use a similar approach to this with the coarse graining and the local feature computation to do something like one of these other paradigms I talked about with the sorry, it's way back here. With the like the interface recognition, I don't know, maybe you could do, if you could identify the interfaces on the nanoparticle, and then you basically try to reverse engineer the protein from that. But I don't think that our specific method would apply in that situation, because we assume the protein is known.
Okay, and the other question is, if you have two, maybe three or four
Mhm.
such nanoparticles, and instead of calling them just nanoparticles, you make them as reactants.
Mhm.
Can you reverse engineer, as you put it, the protein to make an enzyme that would react and cause some kind of effect on these various nanoparticles that have been bound by this protein you've created? Could you make an enzyme?
So your question is, like, if you have two nanoparticles that go into some reaction and one that comes out.
Yeah.
I see. Okay, so so maybe this is related to your first question. Yeah, where if you don't know the protein, then you can't make any kind of prediction. But if your goal is to engineer the protein, then yeah, then you may be able to do it, because then you know the protein, in that you can make predictions. You just need to start with some guess of what the protein is, and then like optimize it from there. So actually, yes, I think that would be applicable. Sorry, I thought you meant, can we predict it? But yes, you could probably use this to optimize some protein structure to increase the amount of
Either create
interaction.
an
Yeah.
enzyme or modify one.
Yeah, I think you could probably do that. I'd like to amend my first answer.
And I guess the third question I just thought of is, if you can do that, does that scare you at all?
No, what did it?
Okay, that answers that question.
Well, I think maybe a scarier one from that angle is these methods that people are developing to generate protein designs. Like, I don't know if you've seen, but there are some methods where basically you are given some protein, and they say, make a protein that binds to this, and this machine learning model will like generate some protein design. Or you can say, like, I have some protein that I want to drug, so I have like this binding region of a molecule, but I want something else to be like attached to it. And you can do linker design like automatically. I'd say maybe those are more immediately scary, because you can just generate them automatically.
Yeah, it's well, it's speculative, but it's also interesting. All right, thank you.
I have a question about your training data set.
Right.
as you know, for the interactions, there is some could be regarded as strong interaction, and sometimes there is also the weak interaction. So, do you classify this kind of interaction or just mess it up is regarded interaction and treating this as if it's two important that interacts, so you regard it as one no interaction, zero, just like this?
Yeah.
Or not?
Yeah, we don't, it's just binary in our,
Binary.
like, so the model makes a prediction between 0 and 1, where it's like the probability of being an interaction. But in our training data, it's just 0 or 1, no in between. We have, so I know that in our data set, we have proteins that we consider to be like different difficulty levels. So I don't know how that is computed. I think it's in the supplemental material of our paper, but that was something Jacob did. And basically, we had these 230 proteins, and we said like we know some are easier to predict than others. So we want to make sure that the model is trained on, both of these. We don't just train it on a bunch of easy ones and then test it on a bunch of hard ones or the other way around. Yeah.
Okay, thank you.
First, fabulous.
No, thank you.
Second, probably just like a clarifying question on your course grading.
Yeah.
So I'm used to course grading having like really hard rules where like, oh, a lysine is n beads with these parameters. This isn't that. This is more like a local algorithmic
Right.
coursing.
Yeah. So the idea, I guess, is that if you use like a standard course grading approach where you are giving like one residue, like amino acid residue gets one bead, then you're sort of, you can think of that as like you have, some specific like beads, one associated with each residue. And the model's more or less just gonna learn that like how to operate on proteins, but because we want it to work on nanoparticles, which you can't do that like one-to-one association. We need it to be sort of random so that the model learns how to deal with chemistry that is not like specific to one amino acid. Maybe this overlaps, two amino acids or something like that to get some like variation in the bead. So yeah, that's my understanding of why we do it. Yeah. Yes.
For your results, can you say a little bit more about how you're scoring it? It was that area under the curve.
Yeah.
But I didn't understand what that meant.
Yeah, so I'll clarify so there are two things that we look at. We look at, well, I guess it's if you think of it as a truth table, there are like 4 things we look at. We look at the AUC inter, which is like the area under the curve of the predictions for like the interface predictions. So we make pair interaction predictions, meaning that we make pairwise interaction predictions, meaning given like 2 structures, where do they interact together? But these methods we're comparing against are interface identification methods. So they don't, they just tell you where on this protein will something bind. So what we do is we take this pair interaction and we convert it to just an interface prediction. And then we compute the AUC for that. So we're basically, I don't know, does that make sense? Basically, we take this, the prediction, so we take the predictions across like all the possible interactions and we say, or basically we convert it to like given this spot on the protein, how likely is it something will bind there? And then we basically compute the AUC over those predictions, which is the AUC inter. The comp means that we are computing this AUC per complex and then averaging over them. And the reason we do that is because some complexes are like hundreds of times larger than others, which biases the AUC. Because if you just average over all the beads and all the structures, you're not equally weighting all the structures, you're sort of weighting them based on size. The AUC pair is looking at all of the pairwise interactions that we are predicting. I guess maybe to make this a little bit clearer, what we do is, okay, so when we make our predictions, we're making predictions like this, like between pairs of beads. But that's what we call, when we're looking at the AUC, the AUC pair, that's what we're looking at, is the predictions for each of these pairs. But when we compare against methods that don't do this, that just look at the protein, basically what we're saying is we're like taking we're taking the predictions for like all three of these predictions, and we're converting them to just a number that says, does this bead interact with anything? And then we just have predictions for the protein, not the nanoparticle. And then we compute the AUC based on that. Is because a lot of these methods are based on the like interface identification prediction problem, where you're just you're not looking at the pairs, you're just looking at one, and then you want to identify you want to identify what region of the protein is involved in that interaction. I don't know, does that make sense?
So I guess, is it like you think of like the area under the receiver operating characteristic, you have a binary label of reads.
Yeah.
You're looking at a ranked list of
Mhm.
predictions, and so you're
Yeah.
trying to look for the enrichment of the true positive label
Yeah.
spheres relative to the other ones.
Yes, yeah. Yeah, so that's the AUC. We compute that for each protein individually. And then we average over all of them, because we otherwise, if you don't average, then some protein that's very large will dominate the AUC, compared to some protein that's very small. So we compute the AUC for every protein individually and then average over that. So each protein is contributing equally to the AUC, I guess.
I guess maybe are there clusters of your data that sort of naturally all look very similar, or are they all distributed? Like, for instance, you have some gold nanoparticles and some.
Oh,
thin
right, yeah,
nanoparticles.
so our nanoparticles, I believe, are all organic in this paper. The concept is more general than organic nanoparticles, but that's what we focused on for this paper. There are, so off the top of my head, actually, I think we have sorry, my mouse kind of disappeared, but oh, here it is. Okay. I believe this is a list of proteins that we had, which is not very, or protein nanoparticle pairs we have, which is not very helpful. But I believe that several of these are molecular tweezers. Several of them are graphene quantum dots. We have some Buckminster fullerene. And I don't remember the rest, but this is in the supplemental material if you want to check and see.
What is number of sites? Number sites?
I believe that's the number of
We
beads.
are at the hour, so I'll make sure that
Yeah.
people know that now. So we're just going to quickly share a comment that Angela had added, where she just wrote a very cool question about the potential of scary applications. Thank you. So with that, if it's okay, I think maybe we'll end here and you guys can.
Yeah. Whoop. Yeah, you have any questions on?